Our next speaker is Dr. Michael Rose, who you just heard mentioned. He's a distinguished professor of evolutionary biology at the University of California, Irvine. He's best known as founder of two scientific fields, the evolutionary biology of aging and experimental evolution. Please join me in welcoming Dr. Michael Rose. You want the clicker?
Thank you.
If you need the slides.
Thank you. Wow. It's very hard to follow some people, and Natasha is one of them. So this will not be fun in the way she is fun. And I'm sorry, I'm a hopeless nerd. So sorry about that. I'm going to be throwing data at you because I don't have an identity without data. I don't have a career without data. I've been working in the aging field for more than 40 years, since 1976. During that time, I have seen a massive swing in work on aging, a swing in which basically people are literally betting their lives on reductionist solutions to the problem of aging, specifically cell molecular solutions. Most specifically, SENS, as put forward by Aubrey de Grey. Now, in fairness to Aubrey, because he doesn't have a background in biological experimentation, I think he's a brilliant man, but he knows not what he does. And today is one of my attempts to save lives by stopping people from doing things that will kill them. All right? So if you don't want to die for the sake of reductionism, then you should pay attention. I'll just mention, let's see if I can find the, do I have a highlight? Is there a highlight laser pointer? Is that the laser pointer? This is the laser pointer. I have co-authors, Grant Rutledge, now with the USDA, Thomas Barter, but to get his PhD on Monday for a scientific breakthrough that unfortunately I can't talk about today, but if you were to come to Irvine in 2 days, you could see that scientific breakthrough. So first I'm going to define what I mean by reductionism in aging research. Reductionism is when you suppose that aging is a physiological process that has just one or a few underlying mechanisms. This might be one mechanism like somatic mutation. That's when mutations occur to your cells in your body. Or cumulative molecular damage. The most famous one of those is free radical damage. And literally in the 1950s, people supposed that either this alone was the sole cause of aging, or that this alone, specifically free radical damage, was offered as the sole and sufficient cause of aging. More recently, Aubrey has increased the favorite number to 7 mechanisms behind aging. Now, you don't actually have to take anybody's word for anything in this field. Let me just try to bring up my talk outline again. Here we go. We now have scientific tools that were unimaginable in the 1950s. They were being built circa 2000, but had not yet been used to answer this very simple question: is aging complex? Okay? So if aging were not complex, we'd find one genomic site or a handful of genomic sites which would affect a handful of transcriptomic differences. The genome is your genotype. The transcript is all the RNAs your genome makes. Then there's proteins. Then there's all the metabolites. Today I'm just going to give you examples from genomics, DNA sequences, and transcriptomics, which is basically RNA sequences. And if you're a fan of reductionism, you really ought to know about DNA and RNA, or you're flying a plane without, knowing anything aeronautics. So As an experimental evolutionist, I have made a specialty since the 1970s of making organisms that live longer. I'm going to show you how I did it. I'm going to show you what the demographic consequences of it are. And then I'm going to look at what that experimental evolution says to us. Now, the key experiment that I became famous for in the early 1980s, it's called various things. Sometimes it's called the Methuselah fly experiment. Sometimes it's called the career woman experiment, not something I endorse because it should really be just simply the career professional experiment, men and women. Normally, this experiment is done in fruit flies, Drosophila. It doesn't have to be done in fruit flies. It has in fact been done with mice as well, along with many other organisms. If you have genetic variation, it always works. Normally, lab organisms are reproduced with a very fast life cycle where we take young adults, we make them lay eggs to start the next generation, we discard those adults, and we complete the life cycle. And this is a fruit fly life cycle. But that is done entirely for convenience. It is instead possible to delay when you allow the adults to reproduce and shift them to later and later ages, much as people do when they go to graduate school, to get PhDs or medical school. And if they're sane and actually do complete their degree, they almost always delay their reproduction a significant amount for very good reasons. There are two really hard things to do in the world. One of them is to get a serious doctorate, and the other one is to produce a child and rear it. Combining them is phenomenally dangerous. If you culture your organisms like this, with delayed first reproduction in the entire population for multiple generations, you are forcing evolution to produce an organism that will live longer. Okay? So what does that look like? So this is just one example of an experiment like that. Here come the data. This is basically a measure of your death rate. The higher up on this graph a dot is, the higher the death rate is. These are data from normal fruit flies which show an aging pattern, this is a log scale, where the death rate shoots up very quickly. It then hits a plateau, which is something reductionism finds hard to explain, but evolutionary biology can. So here's the aging phase for these normal fruit flies. Very fast, very brutal, kills off everybody effectively by this age in days from egg. And this is data from 10 populations. On this graph, you're looking at the pattern of life and death for around 100,000 organisms, all handled together in parallel. These are organisms that we have made evolve postponed aging in the lab. They are not aging initially. They have very, very low mortality rates, and only later does aging kick in and increase the mortality rates to a later plateau. That is the experimental material we are going to use to answer questions about how genomically and transcriptomically complex aging is, because we can determine just what evolution had to do genomically and transcriptomically to produce a longer-lived organism. And the blue ones here live about twice as long. So in human terms, that's routinely living into your 150s or 160s, some individuals living to 200 years plus, to translate from fruit fly into human. So this is just the material we're going to use to answer the key question, is aging complex? This is a whole lot of detail you don't need to worry about from the publication, Graves et al. 2017, where we looked at the genomics across about 1 million single nucleotide polymorphisms in the fruit fly genome. And I'm now going to show you a really staggering slide. Just look at the top row. This is the result of 1 million tests of single nucleotide polymorphism differentiation. The significance levels are up here. They correspond to significance levels of 10 to the minus 100, 10 to the minus 200, 10 to the minus 300. Those are astronomical levels of statistical significance. If aging were genomically simple, you would only see 1, 2, 3, maybe 4 spikes. How many spikes do you think you see? Yeah, thousands. Yeah, there are thousands of spikes there. So aging, or more specifically, to double the lifespan of these fruit flies, which is what we made evolution do, takes the retuning of thousands of sites across the entire genome. Thousands. Okay. Thank you. We can also look at transcriptomic differentiation, and I'll just focus on this line. We sampled the transcripts at about day 21 when the normal flies are dying quite rapidly and the long-lived flies have not even started aging. And you look here, this is the corresponding data. There are more than 660 transcripts which are significantly differentiated, and those transcripts come from all the way across the genome, just like the genomics do. Aging is also genomically extremely complex. Aging is actually very complicated. Very, very complicated. As an aside, we're now working on how to decipher it, and the short answer is we're using machine learning, which many of you know as AI. But I'm just going to ask you the question, given that aging is in fact very complicated, do you think that pretending it isn't complicated will lead to useful ways to postpone your own aging? Are you willing to die for a scientific mistake? You can get plenty more detail. I have a Google Scholar page. I have a website. These are 3 of more than 5 or 6 books I've done on aging. Thank you.